Thank you for coming today. I'm Yoshihiko Fujita from CiRA, a Center for iPS Cell Research and Application. Today I talked about artificial gene switch and its application. Before starting to talking about switch, I'd like to show some pictures. These are the microscopic images of the human cells. From left, 293FT cell, HeLa cells, and iPS cells. And please remember the morphology of these cells. Remember? Okay. And these images are the same cells in the previous slide. Left images are 293FT cells detached from the surface of the culture dishes. And middle and right images are the HeLa cells and iPS cells cultured in low density, respectively. So, as you can see, these images are very different from those in the previous slide. So, as you like this, the morphology of the cells depends on the culture condition. So, as you know, iPS cells promising resources for cell therapy because iPS cells can produce a lot of cell types. In CiRA, we prepared a lot of cell types and use it for clinical applications. For example, neural cells were prepared for Parkinson's disease and cardiomyocytes prepared for heart diseases. But sometimes differentiated cells from iPS cells are heterogeneous, like this. So we have to at first purify the specific cells from unwanted cells. Then we can transplant these cells to diseased organ. If this purification process is insufficient, unwanted cells or unexpected cells may be contaminated and transplanted to our bodies. It is a very serious problem for us. So we have to purify at first. But by the way, how many cells are there in our bodies? It is unclear, but according to some papers, there are about more than 200 different cells. So we have to distinguish at least 200 different cells. How we check the cell type and purify the specific cells? Of course, cells cannot answer our questions like what kind of you. But the smartphone can answer our questions. For example, when we ask who are you, the smartphone may answer, I'm your virtual assistant. If cells can answer our questions like the smartphone, it is very easy to purify the specific cells. The smartphone realizes the answer by circuit and programs. The circuit is consisted of a large number of switches. So our approach is to install switches to enable cells answer our questions. But electrical switch or electrical circuit don't work in cells, so we have to find another alternative molecules which work as switches. When we observe carefully cell behavior, cell can respond to a lot of input signals like circuits. So, for example, cells start to grow in the presence of the specific protein signals called growth factor, and also specific proteins called Fas ligand promote the suicide of the cells. So cell can detect a lot of input signals and determine their behaviors depending on the signals. And natural cells include a lot of switches called gene switch. These gene switch interact with each other and form a very, very complex network. This complex network is called gene circuit. So this is a small portion of the gene circuit, and it is very complicated. So this complicated gene circuit can recognize a lot of input signals like light, heat, small molecules, and proteins, and control the output cell behaviors. So if we install artificial gene switch and construct artificial gene circuit instead of endogenous gene circuit, we can change the input and the output cell behavior. That is, if the installed artificial switch can detect cell type specific molecules and produce fluorescent proteins, we can distinguish each cells by the change of the fluorescent signals. Now I'd like to show the actual molecules of the natural gene circuit. DNA is a very famous molecules, and DNA carries genetic information. And the protein is made according to the genetic information in DNA and work as enzymes, and finally determine the cell behavior. The RNA is similar to DNA, and the messenger RNA, which is one kind of the RNA, copies the genetic information from DNA and transfer the genetic information to factory for protein synthesis. So DNA produces RNA, and RNA produces proteins. And the step from DNA to RNA is called transcription. And the step from RNA to protein is translation. Some proteins control the transcription and translation by DNA protein interaction and RNA protein interaction. This behavior is similar to switch. So we may be able to construct the artificial gene switch by using these molecules. So we focused on the RNA. And we tried to integrate switch system to messenger RNA. Focusing on the translation, the protein is synthesized as follows. At first, genetic information is copied from DNA to messenger RNA. The messenger RNA has a mark of the messenger RNA and encoded gene. The messenger RNA is scanned by translational machinery called ribosome from the mark at the end of the messenger RNA. When ribosome find encoded gene in messenger RNA while scanning, protein synthesis starts. It is important thing that the ribosome scans from the mark at the end of the messenger RNA. Please remember this part. And I'd like to introduce another important molecules, microRNA. MicroRNA is one of the RNA. And microRNA is a small RNA which digests complementary sequence of messenger RNA. MicroRNA recognizes by forming double-strand RNA and digest the messenger RNA at that point. And the number of the candidate of the microRNA is known to be more than 1,800. So this number is more than 200. So if each cell type has specific microRNAs, and if we can detect endogenous such microRNAs, all cells can be categorized by microRNA. So we tried to construct a microRNA responsive switch. Our microRNA responsive switch is very simple. We just inserted microRNA complementary sequence into the messenger RNA. This microRNA responsive switch has a mark of messenger RNA and encoded gene. So we usually use fluorescent protein as encoded gene. This microRNA responsive switch can be translated by fluorescent protein. Ribosome. So, and produce a fluorescent protein, resulting in the change of the color of the cells. In the presence of cell type specific microRNAs, this cell type specific microRNAs recognize this switch and digest switch. Because ribosome scans from the mark at the end of the messenger RNA, so the protein is not synthesized anymore. So we can distinguish the cells which has cell type specific microRNA from others by decreasing of the fluorescent signals. This is an image of the cultured iPS cells, a little bit difficult to see, but there are two types cells in this image. Small and high density cells are iPS cells, and larger cells are differentiated cells. Like this, iPS cells start differentiation while culturing. So it is important to find the differentiated cells to keep the iPS cell quality. It is known that iPS cells highly express microRNA 302, and differentiated cells don't express microRNA 302. So we constructed microRNA 302 responsive switch and installed this switch to iPS cells. This is relative fluorescent images. The translation of our microRNA responsive switch is replaced in HeLa cells because of the microRNA 302. In contrast, the fluorescent signals from differentiated cells are observed. So we can distinguish these differentiated cells and iPS cells. We also constructed several switches which can respond to microRNA 208, microRNA 122, and microRNA 375. These microRNAs are specific molecules in cardiomyocyte and hepatocyte and pancreatic cells. So we prepare these differentiated cells from iPS cells and try to purify these cells by our microRNA responsive switch. For example, microRNA 208 could purify the cardiomyocyte, and the purified cardiomyocyte started heart beating. And the other microRNA responsive switch also could purify the hepatocyte and pancreatic cells respectively. Lastly, I'd like to talk about the future of the artificial gene circuit. Artificial gene circuit consisted of multiple gene switches can detect multiple input signals and determine the output depending on the input signals. So, for example, a genetic gene circuit changes the cell colors in the presence of only two specific input like this, and it also, same gene circuit can promote cell differentiation in the presence of specific four input signals. Like this, gene circuit can make a decision depending on the combination of the input signals. So we'd like to design such a sophisticated gene circuit, and we'd like to develop a technology which can let specific cells take specific action we desire. For example, cell differentiation or cell death. Because I would like to use this technology for clinical applications such as regenerative medicine or tumor drugs. Thank you very much.